Hey, good morning, good afternoon, and good evening, everyone. My name is Michelle Bishop, and it's my pleasure today to introduce Dr. Rich Haspel, who will be presenting today. Dr. Haspel is a current Vice Chair for Medical Education in the Department of Pathology at Beth Israel Deaconess Medical Center and is a Professor of Pathology at Harvard Medical School. He is also a steering committee member of the Global Genomic Medicine Consortium. His work includes leading NIH-funded efforts to develop genomics curricula for medical students and pathology residents, and he's also played a key role in advancing genomic literacy for healthcare providers as a former co-chair of the Inter-Society Coordinating Committee for Practitioner Education in Genomics, a committee which also aims to facilitate interactions among key stakeholders in genomics education. Today, he will be presenting on the history of the ABO blood group system, its role in early genetics research, and its use in defining racial groups through seroanthropology, a practice that was at times misused for racist purposes. By understanding this history and the harm it has caused, healthcare providers can transition now from a race-based to race-conscious approach in transfusion medicine, fostering more equitable and informed clinical practices. So I'll hand the floor over to you, Rich, and I look forward to hearing more about your presentation.
Thank you very much, Michelle, and thank you, GGMC, for giving me the opportunity to talk about this topic, where I've been at transfusion medicine for over 15 years, and I only in the last year or two, only learned about this history of genetics and transfusion and how we're using it in our specialty. And I think it has a lot of application for how we think about genetics and race or in medicine in general. So I don't really have any disclosures. I will just say that some of the terms in the presentation are currently used due to racist connotations. When they appear, I have kept for historical perspective. And while many of my examples are going to be from the United States, I don't think racism and discrimination and issues with genetics and race are unique to the United States. So I'm hoping if you're from another country that it still will be educational. So I only recently realized just how much race is enmeshed in transfusion medicine. And for those who aren't in transfusion medicine, I'm going to show you some of the tables from our standard textbooks. So here's a book a lot of us use called Human Blood Groups. And you'll notice that this glycophorin A and B are associated with the M and S blood group system. There are a lot of blood group systems in addition to ABO. And you can see they categorize according to race. This is from another one of our textbooks where, again, they talk about how things are in white versus black people. And, you say, oh, that's kind of old. But here's a paper that came out in 2024, which is a very important paper about red cell immunization in sickle cell disease patients. But here's a quote. Donor attributes likely contribute to the variation in alloimmunization rate observed. And then the next sentence, donor race was not available due to the retrospective nature of the study. So equating race with genetics and biology. So you'd say, all right, this is in transfusion medicine. What about the public? Well, this is from the New York Times in 2019. Blood can be racially or ethnically specific. So having more blood donors in certain groups can be crucial for saving the lives of patients who share their backgrounds. And then there was a quote from actually someone from the British National Health Service in charge of blood donation. Literally, quote, "We need black blood." This is from the ABB, which is the largest blood bank organization in the United States. Research shows the best match for a blood transfusion is from a black donor for sickle cell disease patients. And this, if you just go to the ABB FAQ about blood and blood donation, they show a list of ABO types based on race and ethnicity. So do we have a problem in blood banking regarding race-based medicine? I would say we definitely have a problem with race-based transfusion medicine. And so what I want to talk a little bit about, and some of you might be familiar with some of these concepts, but to make sure we're on the same page, what is race that is not biological? And what do I mean by race conscious versus race-based medicine and how race-based medicine is dangerous. Why is race so enmeshed in transfusion medicine? And this is this history of seroanthropology, which again, I just really learned about relatively recently. And then I'm going to give an example of race-based transfusion medicine, which is racially matched transfusion, saying, again, a Black patient should get Black blood from Black people, and how that was in the past and how it's still with us. And then again, how can we practice race-conscious transfusion medicine? And part of my goal here is to really look at evidence and what is the data to support certain practices. So what is race? Okay, so race is not a biological construct. And you'll see in the reference below, there was a really important document that came out from the US National Academies of Sciences about population descriptors and genetic research. And this is basically from that. Race is a socio-political system for classifying and ranking human beings according to subjective beliefs about shared ancestry based on perceived innate biological similarities. So you look at someone, the color of their skin, what they look like, and you make judgments about them. The system, and race is not race, it's not scientific. So it varies whatever country or place you're in, how people define race, different racial groups. This has also changed over time. If you look at, in the US, we do a census I think it's every 10 years. So if you look at the categories over time, they change because again, it's not biological. It has been used to promote superiority of one group over another. And what's really remarkable, in the US and actually in other countries, skull size was used as the basis for the idea that race predicts characteristics such as intelligence. Not surprisingly, this was disproven, but this is a paper from a guy named Morton in 1839, where he measured the skull sizes of different races and claimed that Black people have smaller skulls and they're not intelligent because of that. And if you're interested in a book about this, Stephen Jay Gould's a population biologist, wrote a book called Mismeasure of Man about this and other aspects of race and genetics and intelligence. And nowadays, many of you probably know, genetic studies have shown racial classification is now seen to be of virtually no genetic or taxonomic significance. And I'm going to show how that actually comes from, that quote is actually from 1972. Yet we still, in medicine, we equate medicine, we equate race with biology. And equating race with biology leads to bad medicine. So again, just to sort of summarize, race and ethnicity are socio-political, not biological. This leads to typological thinking. I see what someone looks like, and I make judgments about them. That leads to harm, like they're not intelligent or something. Genetic ancestry is something that can be measured quantitatively, but this is very complex and it's important, but it's key to understand genetic ancestry is not the same as race and ethnicity. So this was a paper in The Lancet where I think it's very helpful because it distinguished between race-based and race-conscious medicine. So race-based medicine is when you have racial categories and you use that to infer biological significance. So in one case, you could say, oh, certain races get certain diseases. So you make judgments about what diseases people have based on, for example, the color of their skin. Or you believe race is biological in the sense, oh, it's used in clinical algorithms. Like in the US, we estimated kidney function and we did it differently if someone said they were Black or if they were white. So this leads to racial health inequities. So that's race-based medicine. What is race-conscious medicine? There's no question in the United States that certain racial groups are discriminated against, and that applies to medicine as well. So we need to understand the effects of structural racism and understand how that leads to healthcare inequities and figure out how to reduce those healthcare inequities. So here's an example of race-based transfusion medicine. So there's a phenotype, the Duffy blood group system. It's another blood group system, and some people are negative for two of the big antigens in this system, Duffy A and Duffy B. This originated in sub-Saharan Africa. It provided resistance to malaria, and people move. They migrate. So this is a quote from again, a textbook in transfusion medicine. And it says, in Black people, the Duffy A negative, B negative phenotype in the US is about 0.7, so 70%, but is quote unquote extremely rare in white people and most other races. Population geneticists know that race does not define biology. So this is a heat map of the frequency of the Duffy A negative, B negative phenotype. And yes, it started in sub-Saharan Africa, but then people migrated. So there are plenty of people in the Middle East or South America who have the Duffy A negative, Duffy B negative phenotype. And some of you might be familiar with the term cline, which is when a trait migrates and you see it distribute over time. So how does this cause harm? Well, so genetic traits and diseases do not follow racial divides. So in the United States, there are people with Duffy A, Duffy B negative have lower neutrophil counts. It's completely normal, but they have lower neutrophil counts. And sometimes people think they're neutropenic. They have too low neutrophils and they get bone marrow biopsies. Or in cancer studies, they actually aren't treated because the treatments lead to affect the white blood cells. So they're not given chemotherapy because they think their white cell count is too low. So this in the United States, because the most common population are people of African genetic ancestry, so Black people in the United States are really affected by this, but we can't just assume. There are plenty of other people in the world who have this. So you can't make assumptions that you shouldn't just test Black people for the Duffy A negative, B negative phenotype if you think they have a neutrophil count that is low that you can't explain. And I think one of the best, well, it's not a best, it's a very disturbing example of race-based medicine this is published where there was a Black child who, since they were a little kid, had lung problems. And no one could figure out what the lung problems are. So the kid's 8 years old, and they go to a new doctor, and they get a chest x-ray. And the radiologist, who didn't know the race of the patient, says, who's the kid with cystic fibrosis? So I was taught in medical school that cystic fibrosis only affects white people. But it doesn't. It affects other people. So this poor girl was not diagnosed because her physicians were so immersed in race-based medicine and didn't put cystic fibrosis in their differential. And here's just some other examples of race-based medicine. In the US, there was a EGFR, is glomerular filtration rate. So you would estimate it based on a chemistry result called creatinine. And there was an algorithm that did different things whether you were Black, you identified as Black person or a white person. And it accepted worse kidney function in people who are Black. And what that resulted in is, with changing this to not being race-based, 31,000 more Black patients would become eligible for kidney transplants. In the United States also, pulmonary function testing, lung testing, had a racial component. And only recently did the American Thoracic Society remove that and say how this had caused medical harms for people who are not, for non-white individuals. And this is just a paper in the New England Journal of Medicine, 13 examples from 8 specialties of race-based clinical algorithms. So what does this do? It causes typological thinking and can harm populations. So we need to move away from this race-based medicine. However, we still need to practice race-conscious medicine. We cannot ignore racial issues. There are very disturbing healthcare disparities in the United States. Here is infant mortality. It is twice as high in individuals who would identify as Black than, for example, non-Hispanic white individuals. And these terms are the ones used by the government, so that's where those terms come from. This is a paper on morbidity during childbirth. And you can see again, it's different in people who identify as Black. So there are clear issues in the United States. So we cannot ignore it, but we must understand this is not due to biology. This is due to discrimination and structural racism, which is something we have to remove. Okay, so that's a little bit about race and what I mean by race conscious and race-based medicine. So the next question is, why is race so enmeshed in transfusion medicine? And this relates to the history of seroanthropology. So what is seroanthropology? So I'm going to go through sort of the history of this. And, this is the timeline. But we're going to start with Landsteiner, who discovered the ABO blood groups in 1900. And then 10 years later, Dungern and Hirschfeld discovered this as essentially the first Mendelian trait. So people were very excited about ABO because it's easily measured and it was clearly Mendelian. So at around the same time that they discovered ABO was Mendelian and other traits, people were understanding that this thing of skull sizes or, using physical attributes or physical anthropology to predict characteristics was nonsense. So Virchow, who some of you might have heard of, did a study of 6 million German school children about their skull sizes and showed this is nonsense. Okay, and the idea of Aryans is ridiculous. Franz Boas, who is essentially the father of modern culture-based approaches to anthropology, to say it's not just inherited things, it's your culture. He did a 1911 head size study of children in New York City, and he looked at over 17,000 people. And he said, guess what? Skull sizes are not inherited. They're not purely inherited. They can vary because of cultural aspects, or what we would know today like nutrition and other things like that. So all this time, people were like, this idea of using things like skulls to say, certain people are worse than others is ridiculous. But when it's just, I don't know, something natural about people, they look at people looking different and they say, oh, there must be races. How can there not be? That person looks different than me. They're a different color. So the answer must be genetics. We can use genetics instead of skull sizes to divide people by race. And guess what? ABO blood groups are very easy to test. So that leads to the next step, where the Hirschfelds, husband and wife team, did really the first study in 1919 to try to define race by blood group. And that caused a lot of excitement because people were like, oh my gosh, I can use blood. And then other blood groups were discovered. And they're like, wow, these are Mendelian. We can use it to predict all sorts of things. So by 1939, over 1 million people had been typed trying to prove all sorts of different things like mental illness, you name it. However, people were using it to also categorize people by race. So this was the initial paper by the Hirschfelds, and they happened to be during World War I. They were in a place where there were a lot of troops, thousands of troops that they could do ABO blood group testing on, and there were troops from all over the world. So what they developed from over 8,000 individuals is what they called a biochemical race index, with the idea that the closer you were to sort of West or Europe, the more likely you were to be an A, and the more Eastern or Africa and Asia you got, the more likely you were to be a B. And this was the beginning of seroanthropology. And here's a table from a figure from their publication in The Lancet. And if you go left to right, so the lighter color bars are group A, and the darker bars are group B. And what they try to show here is that as you go west to east, you have less A and more B. Well, they admitted, it's not a perfect thing, but they're like, here's this index to biochemical AB index related to race. And this led to people getting very excited about using blood groups to define race, and as well as other things like diseases and behaviors. but definitely some to define race. And we're talking over 1 million people typed and published. So what is some of that studies that were done between the 20s and 30s for racial classification? So Germany actually had a publication that was actually dedicated to defining blood groups and race. And it was very much based on trying to show Aryan superiority. And, here's a copy of, this is what the journal looked like. Japan, there was the idea of using blood groups to complain Japanese are superior to other people in Asia. In France, there was actually a xenophobic proposal to prevent immigration of type B individuals with the assumption they're from the quote unquote, East and not us. In the US, a guy named Ottenberg, who is actually well known for work in like cross-matching blood and things like that, he developed a classification of human races based on ABO. He didn't claim superiority of one group over the other, but here it is. So this was published in JAMA, which is a pretty big journal in the United States. So what's really interesting, I have some citations here, is this history of seroanthropology is fairly well known in the world of history of medicine. And in fact, I've been working with a colleague at Indiana University who's a historian. They all know about this. However, most blood bankers have no idea about seroanthropology, I would say. So then the 30s to 50s, there were some more mixed messages. There was a book called We Europeans, which was written by a bunch of sort of the beginnings of population genetics and geneticists to try to be a book against Nazism. And what they said is, so they actually included in this book a review of serology to disprove race. And what they said is, hey, Aboriginal Australians have similar ABO as Western Europeans. So how can you claim that how someone looks and ABO are races, exist? So, and this is just a table. You'll notice the old school way of doing ABO was actually Roman numerals. Then there was a guy named Wiener who's actually pretty famous in blood banking for creating another blood group as RH and creating a nomenclature for that. He published in 1946 a division of races. A guy named Boyd in 1950 published in the journal Science a division of races based on ABO and other blood group characteristics. He had 6 races. What you'll notice, though, there's a lot of overlap. It's not like these are very clearly defined. What I found really amazing was some of you might have heard of Isaac Asimov. He wrote the Foundation books. He wrote I, Robot. He published so Boyd and Asimov published a children's book called Races and People, where Boyd listed this use, it was basically about him using this blood group way to define races. So, and again, what you see here is these are pretty similar to other racial groupings. These weren't that unique. And again, not claiming superiority, but all this data, all these tables, these millions of people typed in different places in the world, that is why in blood banking we have all these tables of antigens by race, and we don't really know the history why or the issues and how race is not biological. So the next step in our story is a guy named Morant, and Morant published a book called The Distribution of Human Blood Groups in 1954. And then a guy named Lewontin, who is, some of you might know, a pretty famous population geneticist, actually used Morant's data to disprove biological race. So, and this has been confirmed with genomics. So Morant, what he did, he said, oh my gosh, we have all this data. So let me take all these tables and consolidate them in one place. He didn't divide into races. He just listed things, for example, by the regions where they came from. But he had literally 40 tables. And he even admitted, he said, blood groups show a heterogeneity in the proudest nations and support the view that races of the present day are but temporary integrations. But he wanted to have all this data as a tool for research. And again, this is why in blood banking we have all these tables. And then Lewontin used this data to disprove race. So in 1972, he published a paper, and you can see these are all blood groups. This is the data he used. There were some other things. And then he divided people into racial classifications that were used at that time. And within those classifications, he had populations. So for example, under Black Africans, the populations would be Abyssinians, Bantu, etc. So he compared the genetic diversity between races and populations and within populations. And what he actually found is there's more genetic diversity within a population, these smaller groups, than between populations and races. So actually between races, there is more similarity than within a smaller group population. Confirmed in the genomic era, and this is the quote he had from 1972. Human racial classification is destructive of social and human relations, now seem to be of virtually no genetic or taxonomic significance. So why are we so alike genetically? Some of you may know this, but I just think it's a book called Everyone is African. I just think it's a really good analogy. So imagine you have a bag of marbles. So genetic diversity, well, let's just say genetic diversity in Africa, right? We all came from Africa. So it started with certain common genetic variants, but as time went on, you're going to have new genetic variants develop. And there was a lot of time for that to happen. So imagine these genetic variants, each color circle is a variant, but picture it's a bag of marbles of different colors. So when people migrated, think of reaching into a bag of marbles. What are you going to pull out? If you pull out a few marbles, you're going to pull out the most common traits. So that's why as we migrated and there was development of different groups, we're all incredibly similar because we all came out of Africa, and it was like reaching into that bag of marbles and picking the most common traits. And then within those populations, those smaller populations, there started to be more variation, things like founder effects. So that's why we're very similar. Races, we're all very similar. It's more within populations that there's a lot of diversity. So again, that is a history of seroanthropology. And this is a quote from an article we wrote, which is fascinating to me, especially that I didn't know about this until recently. Antigen typing, blood group antigen typing, went from being used to define race, including in some instances, support of racist ideologies, to a powerful tool in population genetics, and ultimately, and this is what fascinates me, to disproving race as a biological concept. However, we didn't learn that in blood banking. We still use these tables and we still treat race as biological. So what is an example of that? So this is just an interesting quote from a book called Racecraft. And in the United States, we have blood drives related to sickle cell disease and like Sickle Cell Awareness Month. But here's a quote. A tireless time traveler and agile shapeshifter, the blood race equation is back, closed in the good intentions of a Sickle Cell Awareness Month blood drive. So what do they mean by that? Well, until 1972 in the United States, blood was segregated in certain states. And in World War II, blood was not initially accepted from Black donors. And then only blood from Black donors could go to Black soldiers. And that persisted until 1972 in certain states when the last law requiring segregation of blood components was repealed. And this is what blood products used in look like in the United States in certain states. It was listed by race. And they'd only give blood from Black people to Black patients. And look at this. I mean, maybe for some people this seems old, but for me, this isn't that old. These blood labels are from 1969. So, and in fact, so this was purely racist. It was saying Black people have tainted blood. But in 1959, a guy named Scudder, using all those tables, he said, look, the blood group antigens of people of different races are different, and people can make antibodies against those antigens. So based on these tables of race and antigens, we should only give Black blood from Black patients, Black donors to Black patients, based on those tables. So this was an article in the New York Times. Blood expert says transfusion between races may be perilous because of formation of antibodies. And at that time, the blood bank community, in a paper in Transfusion, which is our big journal, said, this is nonsense. This is ridiculous. We shouldn't be using those tables. So you would think this is over, right? The idea of giving blood from Black people to Black patients. Well, I'm going to talk a little bit about something called the Blue Tag program. So what is this return to promoting racially matched transfusion? Here's a quote from the Sickle Cell Disease Association of America. I should say of America. Sickle cell patients benefit most from blood transfusions from people of the same race or similar ethnicity. This is from the American Red Cross website. Blood donations from individuals of the same race or similar ethnicity and blood type are the most effective way to help patients experience sickle cell crisis. This is from the British UK National Health Service. Sickle cell is more prevalent in people of Black heritage, and ethnically matched blood provides the best treatment. This is from American Society of Hematology, our biggest hematology organization in the United States. This is how to best avoid the problem of alloimmunization in SCD. And it's tag blood units from donors of African ancestry to direct them specifically to sickle cell disease centers. So labeling blood that they are from people, well, here they use the term African ancestry, but practically that means people who identify as Black. And again, I showed this paper before, but here's another quote from it. Black donors were specifically recruited by their supplier for patients with sickle cell disease, although other suppliers likely prefer CE and big K type Black donors for selection. And as I mentioned that article in the New York Times, we need Black blood. So I am now going to go through a little bit of evidence that questions a benefit to racially matched transfusion. So first, we talked about back in the 60s, the blood bank community said this was ridiculous, matching people because you're worried about forming antibodies. So, but why did this get reborn? Why did this thing that was discredited in the early 60s now is being done all the time in the United States and UK? So the other question is, are Black patients with sickle cell? So one of the concerns is people are saying Black patients with sickle cell disease are uniquely likely to make antibodies, as opposed to other people who get chronic transfusions. So I'm going to say, are Black patients with sickle cell disease, are they more likely to make antibodies than other people who get a lot of transfusions? And then, just getting right to the point, does racially matched transfusion in sickle cell disease reduce the rate of alloimmunization or people making antibodies to antigens? Which again, if you make antibodies, it's harder to find blood and it can cause hemolysis if you get unmatched blood. So what is the modern basis? I think it can track back to a paper in 1990 in the New England Journal of Medicine. It was called Alloimmunization in Sickle Cell Anemia and Transfusion of Racially Unmatched Blood. So what they did is they concluded that alloimmunization is a common clinically serious problem in sickle cell anemia, and that is partly due to racial differences between the blood donor and recipient populations. And then they didn't though say we should give racially matched blood, but there was an editorial in that same issue. Others have suggested a more distasteful, if potentially effective solution. Only blood from Black donors would be given to Black recipients receiving long-term transfusions, making the search for compatible units much easier. It's time to bring the question out of the closet. Now, it's kind of interesting when you look at this paper. They had a whopping 107 sickle cell anemia patients. They only had 19 control patients. This is crazy. You're comparing such small numbers. And not only that, of the 19 patients with chronic anemia who didn't have sickle cell disease, 8 had aplastic anemia, who might have been getting immunosuppressive therapies, which we know reduce the rate of alloimmunization. So this is the data that was published in the New England Journal that an editorialist said we should give racially matched blood. So I would argue that the data we had, that paper and other similar papers are flawed. So the next question is, are Black patients with sickle cell disease at higher risk for alloimmunization than other populations? And ABB publishes a technical manual that we all use as a major resource. And this is the quote from it. Patients with sickle cell disease have the highest rates of red cell alloimmunization of any patient group, appears to be independent of the actual number of transfusions received. And they cite this study in a journal called Blood from 1990. So I went back to that study, and guess what's in the abstract? the rate of alloimmunization in this group appears to be an explicit function of the number of transfusions received. So there's a real big disconnect here. So let's look further at the data. So in that New England Journal paper I talked about from 1990, let's look at some of the things they claimed. So remember, in that study, the sickle cell disease patients, 30% versus 5% who alloimmunization made antibodies. And they said 50 sickle cell disease patients, 53% made multiple antibodies against a bunch of different blood group antigens. And they wrote the development of multiple antibodies is very unusual in patients receiving transfusions for thalassemia. And then they referenced a few papers. Well, let's look at the paper they referenced. Vox Sang. It was actually, the rate of alloimmunization was 23%, which is pretty close. Not the rate, it was actually the prevalence of alloimmunization was 23%. And multiple antibodies was actually 69%. There was a paper in Transfusion. It was 23% in sickle cell disease patients and 10% in thalassemia, which did not reach significance. And what's most interesting about this paper, they looked at their open heart surgery patients who at that time were getting a lot of transfusions, and the rate was even higher than sickle cell disease. And they wrote, and this is referenced in the New England Journal paper, and they wrote, the risk of sensitization to red blood cell antigens no more contraindicates transfusion in sickle cell disease patients than in thalassemic patients or open heart surgery patients. And there are just recent thal studies showing prevalence of transfusion. And again, compared to the 30% in that 1990 paper. So here's a paper from Saudi Arabia comparing directly thalassemia patients and sickle cell disease patients, and it was very similar. And what's interesting, you look at these rates, they were giving phenotypically matched blood in some cases to some patients to try to, give the same antigen match so they don't make antibodies. So the idea that for example, thalassemia patients make fewer antibodies is not supported by the data. The other problem is all this data is really flawed because we need rates. So, if someone made 7 antibodies and they only got 5 transfusions versus they made 7 antibodies, but they got 100 transfusions, that's different. The 7 after 10 transfusions is much more likely to make antibodies. So you need rates per units, not just prevalence. And I'll show a study about that in a sec. So I do not believe Black patients with sickle cell disease are at higher risk to make antibodies than other groups. So does racially matched transfusion in sickle cell disease reduce the rate of alloimmunization? Well, let's talk about this blue tag program. So by 1999, and this was mainly in the Philadelphia area, in the Pennsylvania, the US, at all blood drives, the products of donors who voluntarily self-identified as Black were labeled with a blue tag that indicated the unit was quote unquote dedicated to meeting the needs of sickle cell patients. The blood was reserved for sickle cell disease patients for 21 days, and we did that until the pandemic hit. Although maybe not be a blue tag, but places are still identifying units from Black people for Black sickle cell patients. So there's a very important study in 2013 that looked at the effect of alloimmunization in sickle cell disease, and here they really looked at rates. So they hypothesized, this is a direct quote, that alloimmunization would be reduced by transfusion with blood selected from ethnically similar donors. And they even matched the blood for the most common antigens that people make antibodies against. So what did they see? Well, they had an episodic, they didn't get transfused as much category. These people got, and you can see, there's an unalloimmunized group and people who made antibodies, and the people who made antibodies got more transfusions, which is kind of logical. And the rate of alloimmunization here was about 15%. And then they had people who were transfused a lot of blood. You can see hundreds of units. And here the rate was close to 60% of alloimmunization. And, well, that's the prevalence. But when you look at the rates, the rate was 0.3 per 100 units transfused. And these were, again, quote unquote, racially matched blood. It was actually higher than other studies where they just took blood off the shelf that they matched for these antigens, but they didn't try to quote unquote racially match. So it was actually higher when you try to do this racial matching. So And again, I'm not saying we shouldn't phenotypically match. There's good data that by phenotypically matching, we reduce alloimmunization, but we shouldn't just think this is a problem for Black people with sickle cell disease or people with sickle cell disease. We should also do it for thalassemia because they get a lot of transfusions and we want to avoid alloimmunization. And this is just the same group kind of published a second study in 2024 where they looked at rates of alloimmunization at different hospitals. And what you saw is at this one hospital, Black donor units were preferentially selected. And what you can see, there were some places that did some extended matching, but still, what you can see is who had the highest rate, the place that was specifically trying to racially match blood. And it also applied to when you looked at a specific blood group, RH. So this to me shows that the idea that giving racially matched blood leads to less alloimmunization is not true. But you might ask, but why? Well, as to quote from one of the papers I just showed, RH diversity in African-American patients and donors contributes to the high rate of RH alloimmunization because it is classic typological thinking. Remember Lewontin, there's more diversity within than between races. Individuals of African genetic ancestry are the most diverse. So are their blood group antigens. They had that head start, right? So when we look at someone and say, oh, look, they're Black, so everyone who Black is genetically similar is ridiculous. In fact, they're incredibly diverse. So skin color does not capture genetic diversity. It's typological thinking. And what I think we need to do, we need to look back to like 1972 in blood banking, where we recognize that race and biology, race and genetics is a bad way to go. So I hope I've sort of shown that these statements, giving blood from Black donors to Black patients is a bad idea. And it's race-based medicine and it doesn't work. And it promotes a way of thinking, typological thinking that's harmful. So we should stop doing that. So again, just to compare a few things, there was racially matched transfusion pre-1972. A donor was, because they were simply Black, was dangerous because their blood is tainted, purely racist things. The Duke Blue Tag program was based, these are not bad people. They were trying to do something helpful. They just had a very simplistic knowledge of genetics where they said a donor simply being Black was safer for recipients. They were trying to help. The intentions are different, but both are false. Both promote typological thinking that the color of one's skin incorrectly translates to other individual characteristics. And remember, color doesn't predict genetic diversity, and it leads to harm. So how can we, well, this I'll just say, I get a lot of feedback from people in the blood bank community, like, oh, the only we have to say to Black donors, you're helping your community, otherwise they're not going to donate. When every other community in the world, we talk about altruism, and the Black community is altruistic too. So this is a kind of, in a way, a discrimination. You're saying people are going to donate because they can help others who look like them? So, a race-conscious approach is recognizing racism has led to underrepresentation of some groups in the blood supply. Outreach based on say we're sorry, but emphasize, yes, we need a diverse blood supply, but just saying we only need Black donors for Black patients is not the way to go. We need to focus on diversity. And I won't go through this, but we published a recent letter to the editor on this of a way that you can promote helping your community, but not saying one person's blood is better for one person than another, but just that we need diversity in the blood supply. So in my specialty, I think, and elsewhere, we need to really, foster change. So stopping race-based transfusion medicine, stop emphasis on antibody frequency tables based on race, stop promoting racially matched transfusions. What we need to do is promote race-conscious transfusion medicine. We need to think critically, educate ourselves about race and transfusion medicine, and teach what we learn, promote discussion. There's a great paper in JAMA. It's a guideline for using race in the medical and ethnicity in the medical literature, which has some really important use. It's guidelines, and it's only a few pages. everyone should be using this when they're publishing a paper, I think. We still need to recognize there are a lot of healthcare disparities, in particular with sickle cell disease, where because of care in the United States, they unfortunately need to go to different hospitals where we don't, for example, have a record of the antibodies they make, putting them at risk of delayed hemolytic reactions. There are a lot of problems with transition from pediatric to adult care. So let's focus on race-conscious things, on the racial discrimination and structural racism in the United States to improve care, not by promoting racially matched transfusions. Now, this gets a little more complex. Currently, there may be situations when race needs to be considered in transfusion. So, for example, this Duffy A negative, Duffy B negative phenotype, if I was going to start unfortunately, our blood supply is primarily people of European genetic ancestry. So if I was going to start randomly screening products, I'd have to screen a whole lot to find Duffy A negative, Duffy B negative. So it makes sense for cost and time, because someone needs a transfusion, to look for units where a person might have self-identified as Black. However, we need to understand why this is not ideal. And we should be moving to genotyping of all our donors and phenotype matching, but not basing that on race. Because the vast majority of antigens that people make, like this Duffy A negative, Duffy B negative, they make a rare antibody called anti-Duffy3. The vast majority of people, the antigens they make antibodies against, there's not really genetic differences across populations. So, all right. So we have a problem with race-based medicine. I'm hoping I went through these different aspects, and I'm hoping in my blood bank community we can start discussing and fixing it, and we can look at our history in blood banking to figure out how to move forward. And this is just my last slide. I specifically want to thank William Schneider, who is he's the historian I've been working at Indiana University, who's published a lot on this. And this paper just has we put together a list of books about and articles about this to kind of learn about, if you want to learn more about it. So anyway, that was my last slide, but thank you for listening and happy to take some questions.
Thank you so much for that, Rich. It was very much thought-provoking and very, very interesting presentation. I want to open up the floor now for any questions that people may have. Feel free to raise your hand or just jump in. Thank you.
Okay, hi, Dr. Ricky Steuerman, Ariel University. And I joined the last part of the talk, so excuse me if I'm not fully in. I was just wondering if you know the rates of sickle cell anemia, have they been dropping in the States? Have people been using PGD for this? Or what are the numbers?
What's PGD?
Pre-implantation genetic diagnosis.
You know, I'm not. I know there's still a whole bunch of people with sickle cell disease. I don't think it's been dropping, and I'm not. I'm sorry. I'm not sure about if that's being used in prenatal diagnosis. So sorry, but we definitely have still a lot of issues with healthcare disparities related to it in the U.S.
Thank you. Any other questions? Maybe while people are thinking about that, I can ask my question. And I'm really going to pick up on what you were talking about at the end, that yes, completely agree about let's discuss this, let's foster change. But I'm curious to hear your thoughts about how we can actually do that, because it feels like it's very embedded in how people think. So it's going to be quite a mind shift for people to change from that.
No, it's a great question. So I can tell you what I've been trying to do is there's a lot. I get, people hear this and they're like, it's something they've been doing. One thing that I can say, I used to be someone who would look at someone's blood group phenotype and go, oh, I bet they're Black. I would say that on rounds because it was just what I was taught. So some people are very, you should see a review I got to a thing we wrote once saying, we have to do this and all that. So, but what I've tried to do is I have several allies in the blood bank community, and we've grouped together and we meet regularly, and we've published, like that letter to the editor I mentioned. So we're publishing, and a lot of it is responding to things that are published, like letters, and saying, huh, you're kind of saying race is biological. Another interesting example in the US, AB plasma is the universal donor plasma. It's less than 5% of the supply. So people have started to saying that we should use A plasma instead, with the idea like, oh, most people are, there's not that many people in the United States that would affect because they're B or A, that are B or AB, right? The problem with that is they're using our blood donor data, which is mostly people of European ancestry, and actually in certain populations are like 30% are B. So we published a letter and we said, you're making some assumptions there that aren't true. We've also recently, we've gone to some meetings and applied to give, educational sessions. So we had one of those recently. I'm on the editorial board of I've reached out to editor-in-chiefs of different magazines to try to incorporate, I think, those AMA guidelines I mentioned, that publication in JAMA. I don't know why every journal in the world isn't incorporating those. It's not rocket science. There's things like, it's amazing when you look at papers. What do they do when they list? Sometimes you need to list race, right? Looking at social, but a lot of them list white first, right? Or a lot of them are using, they don't explain where their racial categories came from, right? Like, there's very simple things we can all do. So it can be frustrating, but finding your allies and then writing and trying to present on it and trying to move forward. But it's amazing to me how enmeshed this is. And some people just, I don't think I'm ever going to convince. But if I could also, the young, like fellowship director, like the younger group who are learning more about this in medical school, I'm hoping the next generation, they can relate to this more than I can. So, but that's a great question.
Yeah, it came to my mind actually that, yes, that medical school seems to be a good place to start because you're then teaching the next generation, but it's the people who are teaching them are probably going to be having these ideas. So it still does need to be a shift in change. But yes, good luck with that. I think it will be quite an effort, and hopefully we can draw on people here to be an ally as well. I think we've got time for one more question. John, you've got your camera on, so I'm wondering whether that means that you've got a question.
I was biding my time, but yeah. I mean, it might be a bit beyond the scale here, but I work a lot with polygenic risk scores, where we can observe quantitatable differential performance, right? But even within the genetic ancestries, that performance tails off at the outer end, or not even the outer end, but along the continuum. So it's tricky, right, again, to try to represent an individual's placement along that continuum. And I don't know if there is an educational project or a recommendation to kind of present where actual quantifiable genetic differential performance exists, how to communicate that, or if he has thoughts around that.
Well, so I am definitely not an expert in GWAS, but I think from what I know, it's a really important example because, people did GWAS from all one population and they claimed it worked for everybody.
Right.
I think the issue with, I think there was a paper in Science or Nature a while back, which was looking at genetic ancestry. And even though you can divide and divide and divide, they decided to divide it by like the classic,
Right.
I think it's like you said, there's a role, and that's why this, if you haven't seen it, this National Academies paper gets into much more detail about population descriptors and genetics. But I think people have to understand who are doing it, that it is a continuum, right? That we are not building these walls that says, and that we're using principles of population genetics, right? Founder effects, migration, things like that. So I definitely think there's a role for genetic ancestry, and look at that, but we have to be just as cautious and not just using the classic, groupings that are racial and thinking that it's a continuum and trying to explain it by population genetics. And also for GWAS, we're learning you can't just, one size doesn't fit all. right?
Right.
But I wouldn't say, oh, let me get a bunch of Black people and a bunch of Asian, but like, you have to understand that Asian and Black, you know, that's incredibly diverse itself. So it's almost like, I guess, and you probably know better than me, you can narrow probably into 1,000 populations, right? Based on,
Right.
so it's a great question. It might actually be an idea for GGMC to think of a way to, you know, to help people with this, because it's a very, and I don't know, and again, I highly recommend that if you haven't read it or anyone else here, that National Academy's report really tackles this from a genetic research environment. But no, it's an important question.
Thank you.
All right.
Sure.
Thank you so much. I'm going to end the questions there and thank Rich on behalf of everyone here. It was a fascinating talk, got us really thinking about things, and hopefully you've got gained a few more allies to help you with this process. I'm going to hand back to Latifa for just closing the meeting.
Just to say quickly, thank you. And if anyone wants to reach out with me with more questions or anything like that, the main thing is we have to discuss this. And I don't have all the answers, but we have to discuss it. So thanks a lot.
Okay, thank you, Michelle, and thank you, Rich, for this insightful presentation, and you all for participating. And just please, I put in the chat box the upcoming webinar session that is scheduled for January 30th at 10 a.m. EST. And looking forward to seeing you, and happy holidays to everyone.
Happy holidays. Thanks a lot.
Thank you. Bye-bye.